let us study about cell wall. It is present only in a plant cell, fungi, and bacteria, while absent in an animal cell. The cell wall is a tough, rigid layer that surrounds the cell membrane to provide structural support and protection. It is mainly composed of complex substance called cellulose, which provides structural strength. We know that the living plant cell loses water through osmosis, resulting in the shrinkage, or the contraction of the cell membrane, away from the cell wall. This phenomenon is called as plasmolysis. To observe this, let us perform an activity. Prepare a slide of Rio leaf peel in water and observe it under a microscope. We will observe the cells with cell wall, cell membrane, cytoplasm, nucleus, and small green granules, called chloroplast. The chloroplast contains a green pigment, called chlorophyll. The stomata cells are also visible. Now, on the same leaf, put a drop of hypertonic solution of sugar, or salt. After a minute, observe it under a microscope. We will observe shrinkage in the cell contents due to plasmolysis. Similarly, prepare a slide of the boiled Rio leaf. Observe it under a microscope. We will find discolored stomata and other organelles. Now on the same slide, put a drop of hypertonic solution. Observe it under a microscope after a minute. You will find that plasmolysis did not occur. This is because boiling kills the cells of the leaf. Thus osmosis takes place only in the living cells, and not in the dead cells. Note that the cell wall helps the cell of plants, fungi, and bacteria to survive in very dilute hypotonic solution without bursting. This happens because the cell in the hypotonic solution swells, building up pressure against the cell wall, while at the same time the cell wall exerts an equal pressure against the swollen cell. Thus, the cell wall acts as a pressure vessel, preventing overexpansion when water enter the cell.